Good afternoon, everybody. Good afternoon. Welcome, President Latimer. The Holy Trinity is with us today, our VPA, Dean and Registrar, faculty, staff, students, Dr. Richard Katch, who will give us a short speech today, and Dr. Testa's technician, who just graduated with us last spring, Nick Rico, and Dr. Testa himself. So what I'm going to do is introduce Dr. Katz first. Dr. Katz is going to overview the internship agreement that we have with Fox Chase Cancer Center, and I think you'll find that it's quite vigorous and potent. So Rich has been a friend of mine for, what, 36 years? So we have a lot of fun together, and we still are engaged with one another. And I would say that most of the interns that went to Fox Chase Cancer Center flowed through Dr. Katz's lab. And they all ended up in very fine institutions after their experience with Dr. Katz. So thanks, Rich.
Well, I don't have to say anything now.
Next slide.
Next slide, please. So this just outlines the details of the agreement between Fox Chase and Chestnut Hill that was established in 2015. And you can see that this was meant to provide a conduit for talented students to engage in an intensive laboratory experience. So I'm supposed to say next slide now. So this just summarizes all the students that came through my laboratory. There were other students at Fox Chase. And so starting actually in 2023, I co-mentored Kelsey Haw. And I'll detail like the accomplishments of all these students as I go along. But then in 2013, I had Kelly Dunleavy. And this was about the time the formality between Fox Chase and Chestnut Hill began. So my first official student in the program, I guess, was Jason. And then we've gone on to Jade and Maggie. And most of you know Mo. Mo did a volunteer in our lab. And Sam and Aubrey. And it turned out Cheyenne Gilly applied for a technician's position in my laboratory. So that was kind of a coincidence, but it was wonderful to have another Chestnut Hill student. And I also had Chow for a bit. Next slide. So after mentoring all these students, it became quite clear that there was a consensus of qualities that I can very comfortably describe, that the students from Chestnut Hill had strong knowledge base, they're very curious, which is important in science, they're hardworking, and they became owners of their project, which means that the student can come to the mentor and say, I think you were wrong, I think we should not do that. And that's when you know that things are going really well. And so I had that experience with many students, and they were also just generally kind-spirited and generous. Next slide. So I just wanted to outline the kinds of projects very briefly. A lot of cell imaging, and we're working with two proteins, kind of structure-function projects, and these hadn't been worked on before, but they have very important roles. So it gave the students an opportunity to ask these questions about the unknown, because these proteins had not been studied. Next slide. So I already mentioned Kelsey, so I co-mentored her on this, actually a virology project. And then she went on to Yale for a PhD program in virology, and now she's a scientist at Sana Biotechnology in Cambridge. So next slide. And this is Kelly. So Kelly had a first author publication. Actually, this was three first author sharing publication with Jade Wilson, another Chestnut Hill student. And so Kelly went on to get a master's from Penn, and now she's a senior medical writer. Next slide. And again, this is Jade. So Jade and Kelly shared first authorship on this paper, and Jade is now in the Temple MD PhD program. And now she's doing her PhD at Fox Chase, so I get to see her all the time. And she's in the laboratory of Jonathan Wetstein. Next slide. And then Jason, so Jason was involved in 2 publications, and he recently completed his PhD at Temple, and he'll be doing a postdoc at Fox Chase, so I'll get to see him a lot also. Next slide. And lastly, Maggie. So Maggie's in the Temple PhD program, and I have a manuscript I'm working on that highlights Maggie's work in the lab. And next slide. And then, as I mentioned, I had China as my tech. Samantha did a great job, and she's now a nurse, and Chow was in the lab as well. And it turned out Cheyenne is now has a permanent position at Fox Chase. So next slide. And this is just composition showing mingling of the Chestnut Hill students with my other laboratory staff. So I'd like to thank Joe very much for giving me this opportunity, and it was just been a wonderful experience working with Chestnut Hill students. Thank you.
Dr. Latimer. You're welcome.
Good afternoon, everyone. So what a lovely event, Biomedical Distinguished Lecture Series. Let's start by acknowledging and just being so grateful and glad that Lakshmi Atchison is back. Let's give a round of applause to her. And Ken Soprano. Lakshmi, as you all know, ran the program for so long. And an extra special thanks to Joe for taking over the baton and bringing to us what I know will be a really exceptional speaker in Dr. Joe Testa, Professor and Carol and Kenneth Wegg, Endowed Chair in Human Genetics at Fox Chase Cancer Center. And so lovely to hear about the amazing collaborations between Chestnut Hill College and Fox Chase. Those opportunities for mentorship and experiential learning is obviously what sets the stage for them to go on to have exceptional careers and advance their education. So I had the opportunity to speak briefly with Joe about his groundbreaking work in mesothelioma and cancer, and what really struck me in terms of his work, other than my lack of pronunciation skills, is the fact that we share a background of coming from either first generation or second generation college students, and by virtue of that, coming from a background where our parents and our grandparents really were blue-collar workers that were exposed to asbestos and many, many other toxins that lead to cancer and other disease. And it really became clear talking to Joe that he had an enormous passion, and the motivation for the excellent work that he's doing is to continue to reach out to populations at risk and figuring out ways to prevent cancer, and if they are at risk for cancer, intervening early so that they will not develop cancer, and that's just critical work. And I think it's very consistent with the mission of Chestnut Hill to serve the underserved. So with that, I will bring Joe back to the stage. Thank you.
Thanks, Dr. Latimer. Oh, it's there. All right, so welcome, Dr. Joseph Testa from Fox Chase Cancer Center. Could I have the next? Just take a look at this picture. So I put this, no, it's not there, but it's a horse of a different color, so to speak, the architecture between Fox Chase Cancer Center and obviously Chestnut Hill College. Next slide. So this is part of our property here at Chestnut Hill. This is our main building for guests who are not familiar with the college. This is St. Joseph's Hall, and we're actually in St. Joseph's Hall. Right there is the rotunda where we have these great events. And there is the rotunda there. Next slide. So as I was thinking about this lecture, I wanted to kind of ferret out how holding these lectures fit nicely into our mission and values. So I've highlighted many of them in blue. So pursuing academic excellence through interdisciplinary learning, offering challenging academic programs with a global emphasis, and that's clearly the case with Dr. Testa's work, providing opportunities for self-discovery and personal growth, and that's what the students are doing as interns at Fox Chase Cancer Center. Just relationships is one of our values, and so we live this by honoring the dignity of all persons and fostering mutual partnerships, and so I regard our partnership as being very valuable with this college, with Fox Chase Cancer Center. Celebrating one another's potential and achievements, and that's clearly, Rich outlined that, how important it was for those student interns to experience research, high-profile research actually, and it got them to very significant institutions and careers. Cultivating lively forms for intellectual and cultural pursuits. So this campus is quite diverse, and so I would say the personnel at Fox Chase Cancer Center. So it's a lovely relationship we have with them. Expanding perspectives through mentor and peer relationships. Obviously, Rich took on most of the interns, probably, I think, 10 in total over the years, and probably had another 8 in various other labs at Fox Chase Cancer Center. So the mentors or the scientists that I send these interns to are truly remarkable and very kind to our students and give them just a great hands-on experience. Right, Nick? Yes? Okay. Next slide, please. So I do want to thank Rich for the introduction. Not only that, but our relationship goes back about 36 years. I actually worked in the lab of Anna Marie Scalca, who is Vice President for Research at Fox Chase Cancer Center. Lovely, lovely scientist. But I would say that really, ultimately, Rich, you were my mentor, day-to-day mentor, so I really appreciate all the technologies that you taught me. Glenn Real was part of this official articulation agreement that we established with Fox Chase in 2015. Next slide. Bill, you already thanked Lakshmi, so Lakshmi, thank you very much for doing this series for 30 long years. Having experienced it now over the last 8 weeks, I truly appreciate what you've accomplished over 30 years. I'm ready, no, I better not say that. But along with Lakshmi, this is when she was 13 or something. So here's Lakshmi with her husband, Michael Atchison. I want to thank Michael as well, because Michael's at the University of Pennsylvania, and he also served as a conduit to bring many significant scientists, even Nobel Prize winners, to this campus through the biomedical seminar series that Lakshmi fostered for such a long time. Next slide. So actually, Bill, you introduced Joe quite well, so I won't go through all of this. He did get his undergraduate degree at Fordham and did his postdoc at the University of Chicago. And many of you have the flyer, so I won't bother to go through the detail of this. I did see Joe's presentation, so I think it's very important that we hear from him about his studies with asbestos exposure that drives the formation of mesothelioma. Thank you, Joe. It's your turn.
I feel like I'm cutting off the people over here, though. Is that? I'll try to move once in a while. I'm putting a watch here because Joe said if I go past 1 hour, he will be quite upset. Anyway, it's a pleasure to be here. It's an honor to be at Chestnut Hill. It's such a beautiful campus, and it's a beautiful day to be here. I want to thank President Vladimir for inviting me, Joe Gokowski for bringing the news to me about, about a month ago, and Lakshmi, who I've known for many years, and for all of the attendees, students, and faculty. It's my pleasure to have met you this morning, and for others I'll see later in the day. I look forward to that. So I'm going to be speaking to you about 4 things that are listed here: asbestos, a carcinogenic mineral fiber, genetics, both the genetics in humans and mice, and about the lessons we've learned from mouse models and from human cancer specimens. So this is Fox Chase, which was founded in 1904. It was actually out in Lankenau that it was founded, and it moved up to our current address in Fox Chase area, not far from here. Although I think the way that Nick Rico took me here was impossible. It is a small campus with 2,200 employees. It started with this building here. It's a lot longer now, but it was a small campus with, at one time, only about 30 faculty, and 8 of them were in the National Academy of Sciences, which is an incredible ratio. We had 2 Nobel Prize winners in this small campus. Now it's a quadrangle, beautiful area in the center that we often go out and have lunch at. And there's quite a number of employees in the clinical side as well as the basic research and population science areas. I think I'm going to put my glasses on. When you get old, these things happen. I want to at least tell you that Fox Chase is nearby, and we do have research opportunities for undergraduates, as you saw earlier. I'm hoping that Nick saw about publications and about going on and getting his PhD and doing well, or his MD. You can see here from Chestnut Hill to Fox Chase is not that far. And we welcome students in the summer as well as during the year. And Joe had asked me if I would present a brief history of Fox Chase. These are some of the major discoveries that were made at our center over the years. The earliest discovery that was of incredible importance, although it never got a Nobel Prize, it was the discovery of what's called the Philadelphia chromosome. It was this tiny chromosome that seemed to have a deletion. It later turned out to be found to have a translocation. It was called the Philadelphia chromosome because it was found right here in Philadelphia, and it was due to a collaboration between David Hungerford at Fox Chase, who was a graduate student at that time, and Peter Knoll, who was a young resident down at University of Pennsylvania. And that eventually that led to finding out what was at the breakpoints. It turned out they later found the product of that rearrangement caused a certain protein kinase that could be targeted for therapy. And people used to die with this disease, chronic myeloid leukemia, within about 3 to 5 years. And now people like Kareem Abdul-Jabbar, the basketball player, are alive 20 years later. And instead of advertising about his leukemia, now he's advertising about something for, I don't know, what it is. AFib. So he doesn't care about his leukemia anymore. So that's how far we've come. Another discovery was the discovery of the hepatitis B virus, which it used to cause millions of deaths due to liver cancer in China and Africa and Egypt. And Dr. Barry Blumberg identified the virus and developed a vaccine for it. He probably saved millions of lives. Also, he got the Nobel Prize for that work. There was a development of chimeric mice at Fox Chase. These are called transgenic mice, where you make mutations in these mice to make their genome kind of like something you see in a human. And she received the St. George Award, which is a very prestigious prize. There was also another Nobel-winning prize by Ernie Rose for his work in the 1970s to discover essentially the ubiquitin-mediated protein degradation system, what that means for all of you who don't understand that particular terminology. It's sort of like your garbage disposal. It takes proteins that are no longer needed, puts a tag on them, and that tag says, get out of here. And it gets degraded. The two-hit hypothesis, and actually a theory of cancer causation, was identified by Al Knudsen, and this was developed to explain the relationship between hereditary and non-hereditary forms of cancer. And it also predicted the existence of what are now called tumor suppressor genes. These genes are supposed to normally suppress cancer, but if you mutate them, you can end up getting a cancer to be started or promoted or initiated. He won the Lasker Award for that. It's kind of the American Nobel Prize. And he also won the Kyoto Prize, which is a very, very prestigious prize worth about $500,000. Mel Bosma developed SCID mice, severe combined immunodeficient mice, which model a disease that's seen in humans. These mice are used all over the world for various studies. And I was a part of a cloning and characterization of AKT1 and AKT2 oncogenes with Alfonso Bellicosa and Phil Sickless in the early 1990s. Well, something's going wrong here. Anybody can help if it doesn't move. I'm pushing the button.
Greg, can you advance it?
Okay. So this is a year before I arrived at Fox Chase. This is Fox Chase around 1988. These are sort of like the Yankees, and back in the day, the Murderers Row. All of these individuals here were members of the National Academy of Sciences. Barry Blumberg and Ernie Rose are Nobel laureates. B. Mintz, Al Knudsen, and Bob Perry. These were our senior vice presidents of the 3 divisions at Fox Chase. This established, they were one of the 4 cancer centers in the United States that had the idea that you have to have good basic science, good medical science, and importantly, good population science. Many cancer centers waited for years to get good population science. This means epidemiology, cancer control, chemoprevention, and so forth. Fox Chase had to train all these people to go out and become directors at other centers, and now there's about 70 centers that have comprehensive cancer center status. And you can see that at Fox Chase, we memorialized this by having these 3 diamonds with the, supposed to mean with the circle here, they're intertwined, they're interchanging. And these are made from Italian marble, showing the 3 divisions that were run by Ann Scalco, who trained Joe Katz and Rich Katz and Joe Kukalski, Bob Comas, who's deceased now, and Paul Engstrom, who was renowned for training all the cancer center directors around the country. So now I'd like to turn to the main part of my talk, which is about asbestos and erionite, which is another carcinogenic form of asbestos, a form of mineral. It's more carcinogenic than actually asbestos. There are 2 main kinds of asbestos. One is called serpentine because it looks snake-like. It's curvy, flexible fibers. It's used for many different things. All of these contain silicates. So you can see silicon dioxides and trioxides and so forth. So these are woven into fabrics. They're still being used today. This group is still being used today. About 95% of all the asbestos is serpentine. It's in vinyl floor tiles, adhesives, et cetera. You can see here, even putty and caulk, when you use that, be careful, because you should wash your hands and be careful. And when you're sanding it, be very, very careful, because if it goes in the air, that's when it's most likely to be carcinogenic. If you wet it down, it's less likely to be carcinogenic. Amphibole types are crucidolite. There's actually 6 types. I only list 3 here. The blue type is crucidolite. I'll show you that in a moment. Amosite is brown. You notice that they have iron, Fe, iron ions in them as well. But again, they all have silicates, and these are straight needle fibers. They often have these found in older buildings, and you've probably all been seeing over the last few years in a number of Philadelphia schools, like I think Frankfurt High School recently had part of the building was closed down because of asbestos in the schools, and especially in the basements. It's a big problem for these schools in Philadelphia that are very old, because we didn't start to ban this kind of amphibole asbestos until the 1980s. So why do we use this? Well, asbestos is a fantastic insulator. It's just absolutely fantastic. It's used widely for insulating material prior to the U.S. Government restricting it in the early 1980s. Today, its use is largely regulated in Europe and the United States, but it's almost totally unregulated in Russia, China, and India, where it's consumed in large quantities. Its exposure to asbestos has been linked to various benign and malignant diseases. Benign diseases include plaques, like one of my uncles had, asbestosis, like one of my uncles died of, diffuse pleural thickening, and effusions that can sometimes surround the lungs, and sometimes people, I know one asbestos worker I worked with told me he was walking on a golf course with a colleague who heard this slushing sound, and it was the slushing of the fluid around this guy's lungs. And then malignant diseases include mostly mesothelioma and lung cancer. So these are the populations that are at risk since the 1940s, when we started to use this commercially. Shipyard workers and naval personnel around the cannons of ship cannons. You have a lot of asbestos because it gets so hot. You need the asbestos to keep it from starting a fire around the cannons. Auto mechanics, brake lining repairmen, workers involved in removal of asbestos, construction workers, insulators, roofers, furnace boiler workers, and electricians. My uncle was exposed as an electrician because you have to make asbestos to go around the wires to connect them to the furnaces and so forth. And he was often mixing the asbestos, and the powder would fly up in the air, and he was breathing it. And get this one. This is amazing. 1952, when people were first understanding that cigarettes could cause lung cancer, I remember Kent Micronite cigarettes. I'm old enough to remember that. Not in 52, because I was only about like 5 or 4 or something like that. But somebody had the brilliant idea, well, let's fix that by putting crucidolite. Remember the blue asbestos? Let's put that in the filters. So we'll cure you from getting lung cancer, but we'll give you mesothelioma. And don't think that you aren't possibly exposed even here, because the Wissahickon River probably runs pretty near here. And up here, this is a Google map of 2 EPA Superfund waste sites in Ambler, PA, which is probably not too far from here. One is, this is the town of Ambler, and Beaurit is here, surrounded by that yellow, and this is the asbestos pile site, which is surrounded by that red. And this Ambler site grew up as a company town for one of the nation's largest, I think it was called Keesby in Madison, one of the leading manufacturers of asbestos products until the mid-'80s. And the company kept on depositing waste at 2 sites until the 1980s, with the total, get this, total waste volume was estimated to be 2 to 3 million cubic yards. So this, by this, 2 to 3 million yards piled 20 to 30 feet high over this entire area. And next to it, within like a little bit more than a mile, you have Wissahickon High School and Upper Dublin High School. And somebody decided at Penn, a friend of mine named Trevor Penning, to look at this area. And you can see this is before the EPA, Environmental Protection Agency, decided to look at this area. And this is asbestos, this white stuff. This is the serpentine type, the white asbestos. This is piled up in one area at the Boritz site. And at that time, they looked at 3 different counties, and around the Superfund site, they're expected to have 9 cases of mesothelioma. Instead, they had 3 times that number, 28. And guess what? Guess where you are, right here, 4 miles south of the Superfund site. Not to scare you, but none of us can escape it. Anybody who lives in the industrial world is exposed to asbestos. But here's something interesting about that Superfund site. Look how ridiculous we were in the past. This is white gravel. It looks like white gravel. That isn't just like an overexposed film. That's white. That's an asbestos pile. Later on, the EPA remediated it, and they didn't want to put it like on trucks and send it through Ambler and send it past your college and stuff. They decided if they did that, it's likely, even if they covered it, that some asbestos is going to come flying out and into your homes and stuff. So what they decided to do was just bury it with about 18 inches of dirt. It's okay until a tree falls over and then the asbestos flies up. But anyway, take a look here. Here's a basketball court. I don't know if you can see that well, but there's a basketball court right next to this stuff, plus there's swings here. So children were playing right in this area. And now we're more careful in the United States, but in some countries like India, China, and Russia, asbestos is like people have written a book, the asbestos time bomb. They expect in usually it takes about 20 to 30 to 40 years before people show the symptoms of asbestos-related symptoms. And you can see here, people are playing with this asbestos. They're working with it as if it was nothing. No regulations, no gloves, no masks. And look at this kid here. Look at this kid. He's eating right next to where somebody's cutting asbestos, where the sawdust from the asbestos will go right into his food. And we don't really make asbestos. It's not man-made. It's in the ground. And another kind of carcinogenic mineral fiber is called erionite. And you can see it has a very long formula here, but again, it's a silicate. This one has aluminum in it, but again, another silicate. It's more carcinogenic than even the worst asbestos, chrysolite. It's naturally occurring fiber found in Cappadocia, Turkey, which is in central Turkey. Some people love this area because you can fly these balloons over the area, and it's beautiful because you have all these what are called chimney ferries, and you can fly over this. It's such a gorgeous area. It almost looks prehistoric, like on the moon or something. But you have these bundles of fibers. They really like water, but then it can break up into these. It looks almost like a toothpick, and if I'm reading it right, it's about 7.5 microns long, so they're very tiny. They can get into your lungs, and it's not just in Cappadocia, it's also in some parts of the United States, like in North Dakota. there's areas where they've taken from hills and they've used it to build roads. And they have a picture in nature of kids in a bus waving out the window as they're driving over a bus over these roads, and the fibers are flying up in the air into the bus's windows and so forth. So in Cappadocia, this was actually formed by volcanic eruptions over the whole region. And it's covered with lava and a dense layer of volcanic ashes that contain erionite that formed on the surface. And then there's 3 small villages there where it's incredible to believe, because we only have 3,000 cases in the United States of mesothelioma. There, 40 to 50% of all deaths are due to mesothelioma, because they're basically living in houses made of stones that contain this mineral fiber. And if you look very carefully here, here's the villages with these ancient rock formations and these caves that are pointed out here. You can see one here and another one here. They would dig into this because it's such soft volcanic tuff. They can dig in there and make their houses right in there. And in recent years, they learned to, you can see these look like they've been shaved. They shaved them to make bricks or blocks, and they used them to make these houses here. But that means their whole house, from their footing to their ceiling and their walls, are all this kind of erionite. So it's no wonder so many were dying. So before I show you what mesothelioma looks like, a little background. 70 to 80% of, there's 2 kinds of, actually there's 4 kinds. Anyway, there's 2 main kinds of mesothelioma. One in the chest area called pleural mesothelioma. One in the abdomen, it's called peritoneal mesothelioma. 70 to 80% of the ones in the pleura or chest area are caused by asbestos. There's about 3,000 new cases diagnosed annually in the United States. There's about 6,000 or 7,000 new cases of asbestos-related lung cancers in the United States. Usually these are diagnosed in advanced stages. People will come in who are already stage 4. There's not much you can do about it. And the median survival is usually less than a year. With the best therapies, it's about 18 months. The median survival from the initial exposure, the median latency from initial exposure to the disease development, where you see symptoms, it's about 30 years, ranging from, actually it's about 15 years to, it can be as long as 60 years. So there's this really long latency. But less than 5% of even the heavily exposed people, like asbestos miners, people who go down into the mines to dig out asbestos, only like 4 or 5% of those people will ever develop mesothelioma, which suggests that something else must be playing a role here, like there must be genetic factors that protect some people or that make some other people more susceptible. So this is basically this long latency from initial fiber penetration that might transform a cell, which can happen within a few weeks. There have been some people who get mesothelioma that worked one summer or a few weeks in somebody's attic, repairing some people's attics where they had asbestos in the attic, and they never were involved with any asbestos work for the rest of their lives, and they developed mesothelioma. So just a short period of time can be enough. Then you can be dormant for 20 to 40 years. Then something is the final switch to activate it, and from that time to death can be like less than a year. So this is what the disease looks like. This cartoon shows all this white stuff here on the diaphragm, along the outside of the lung, on the parietal area, along the chest wall. That's all mesothelioma. In here, you might be able to see that it's blackened. That's asbestosis, where all the lung tissue is destroyed, so a person is trying so hard to breathe, like my uncle was on an oxygen tank at the end just to try to breathe because you destroyed all the lung tissue. And here's what it can look like in an autopsy photo. All this white area is all mesothelioma, and this is where the heart would be. And you can see the white even around the heart. That's when you just have it around the heart, it's called pericardial mesothelioma. And imagine it started from this single cell layer. This is a normal visceral pleural surface with the mesothelial cells. The surface is a low cuboidal cells, and below here is all like connective tissue and lung alveoli cells. But this is just a single layer. That is so thin, it's thinner than a piece of wax paper. Imagine it goes from that to a tumor that has $100 trillion to a billion cells. Imagine how many divisions you had to have to get from here to there. And how does that happen? That's what I'm going to focus on the rest of my talk. By the way, this is, if you take these cells, and we do this with mice, we grow them in culture, they look like a sort of a monolayer, cobblestone looking cells growing on the surface of a cell culture dish. Unfortunately, this doesn't show up that well on my computer. It looks okay here. This was from a New England Journal of Medicine article written by Bert Vogelstein and Ken Kinsler at Johns Hopkins. They published on the path to cancer, 3 strikes and you're out, like in baseball. To see it a little bigger, you can see what they have here is 4 different cancers. On the left is melanoma, then pancreatic cancer, cervical cancer, and colorectal cancer. And the point to show 4 cancers is just to say, when you have the breakthrough phase, there's 3 phases, breakthrough phase, the expansion phase, and the invasive phase of a tumor. For each tumor type, there might be a different first hit, first gene that's involved. In the melanoma, for example, it's BRAF. In pancreatic cancer, it's KRAS. So it doesn't matter. You just have a first gene that's mutated that might be specific to that tissue. Then you get, and what happens is the cell stays right there, but it's screwed up. It's mutated. Then you have an expansion phase where you still don't invade this, what's called the basement membrane. Here is like, let's say this was lung cancer. This is the lung, and this is that lining. Or let's say this is the lining of mesothelium. It doesn't invade into the lung. It's just out here. And then it can grow into a benign tumor here. This is when you have a second hit, all right? And for this tumor, for melanoma, it's TERT. For KRAS, it's CDKN2A. This is also involved in mesothelioma. Then what happens when you finally invade through this basement membrane, here's that membrane that's going through now, you get other genes that happen, like P53. And you can even get so far as to go into a blood vessel, like it's showing here, and it can go someplace to another tissue, like a lung cancer can go to the brain. So cancer causation is essentially typically involving both genes and the environment. Causes of cancer risk include carcinogenic exposure, like in cigarettes for lung cancer, for mesothelioma, it's asbestos. Individual susceptibility can be if you have a germline mutation that you inherited from your parents, that you have in all your cells. Like, I'm sure all of you have heard of BRCA1 and BRCA2, that women who develop breast cancer can inherit from their mom, and the daughter can get breast cancer because they have in their germline in all the cells of their body, they inherited a mutation in a gene called BRCA1 or BRCA2. Well, we have those now. Our lab has found that you can have that in mesothelioma as well. So here's what happens. Let's say it's a normal person that doesn't have a germline mutation. You'll have a normal cell, then you'll get a first mutation in one of those genes. Things are fine, you don't have cancer, but you have that first breakthrough. Then you have a second hit, and now you might have, that second phase, and then you have a third or fourth mutation, and now you've got invasion, and it forms a malignancy. And this is how I started in cancer research. I started at the microscope, like my mentor, Janet Rowley, who won about 60, 70 awards, including the Lasker Award. She did what's called karyotyping. We all have 46 chromosomes if we're normal. We have a pair of each of the chromosomes. One chromosome came from the mom and one came from the father. So the sperm gave maybe this copy of chromosome 1 and the egg gave this copy of chromosome 1. So we have 2 copies of everything in a normal cell. And a woman will have 2 X's, can I say that? And a male will have an X and a Y. And in this cell, it's from a mesothelioma patient. And you can see these arrows indicate that there are a lot of abnormal chromosomes. 1, 2, 3, 4, 6. So there's about 10 abnormal chromosomes, plus a missing chromosome 2, a missing chromosome 22. And you can't tell it because you don't have the experience, but my cytogeneticists can tell that there's a tiny deletion in here on chromosome 1. There is a tiny deletion on this copy of chromosome 3. The 3p means the P is the short arm, 21 or 2.21 means a chromosome band. We've lost both copies, both of these. Chromosome 9, the P arm, the short arm is abnormal, so we've lost the short arm of chromosome 9. And then we have what's called monosomy 22. We've lost 1 copy of 22. And if we ended up looking at, I don't know, maybe 60 different mesotheliomas over the years. The tumors as well as cell lines derived from the tumors. And what we found is that these consistent deletions are found in mesothelioma. Chromosome 1, P22 at that band, 70% of the cases. Chromosome 3 at that band, 60%. Chromosome 9 is the most common, was 85%, and chromosome 22, 70%. These are not in the normal tissue. These are only in the tumor cells. So here's Al Knutson's 2-hit theory in a simple cartoon. For tumor suppressor genes, I'll call them TSGs. So what you have is you have 2 normal chromosomes here, 2 normal genes. These little things here indicate that these are normal genes, and their normal role is to prevent cancer. And if you kind of make the analogy to a car that has front tires and back tires, that this would be able to stop and not hit a wall. If you get this thing here is supposed to indicate that you get a mutation of one of those 2 copies of that gene. So you lose your back brakes, but your front brakes, if you're not going too fast, might be enough to stop you from hitting that wall. So that might even occur in a person who's susceptible, like a person who has a BRCA1 mutation. It might be enough to stop you. But then you might have a deletion of this other chromosome, and now in mesothelioma we see losses like this of chromosomes 3, 9, and 22. So we know that that is deleted, but what's the mutated gene on the other side? That's what we spent a long time, and I won't, go into those details because it's too detailed. But we wanted to see what was on the other copy. What was the mutated gene there? So what we did was we had various strategies used to discover the critical tumor suppressor genes that are deleted on chromosome 3, chromosome 9, and chromosome 22. For chromosome 3, we did something like Dr. Acheson did. We did loss of heterozygosity analysis, where you're looking for, you're targeting a certain region like chromosome 3p and trying to find out what's the smallest region of overlap to say where might the gene be. And we also later did family studies. For chromosome 9, we did homozygous deletion mapping. For chromosome 22, we said, well, we're losing a whole chromosome, so we're not going to be able to do LOH analysis, loss of heterozygosity. So what we'll do is, if anybody finds a gene that's a tumor suppressor gene in that chromosome, we'll try to see if it's mutated in mesothelioma. And we had basically good fortune. We got lucky.
Joe, are these in cell culture? Are these chromosomal anomalies you see in cell culture or in tumors in vivo?
They were present in tumors in vivo. Whenever we couldn't get, whenever we could get a tumor and a cell line, we'd always validate that we could see it in the tumor. So for chromosome 9, it was kind of interesting. I'm losing my battery here, but here is CDKN2A. I mentioned that earlier. This also happens in melanoma. You have a locus on chromosome 9 that actually codes for 2 genes. It's very unusual. If you look at this locus, oh, you got a good one. Okay. Okay, so there's exon 1 beta, exon 1 alpha, exon 2 and 3. One of the products of this locus is up at the top. It takes this orange area of exon 1 alpha, 2, and 3. This other product takes from exon 1 beta and exon 2. It doesn't take anything from exon 1 or anything from exon 3. So the only thing that overlaps is right here. Oftentimes, when we see a deletion, when we did homozygous deletion mapping, we saw that there would be a deletion that would encompass exon 2. So that was bad because that means we're losing both gene products. And that's really bad because P16 is involved in RB cell cycle regulation, and P14ARF is involved in the P53 pathway. So we're losing we're affecting 2 pathways that are really important in cancer. So P16, we found that, I'll try to speed it up a bit, that, well, bottom line, let's go to the bottom. 100% of the cell lines, we basically had to do this with cell lines because if you do it with primary tumors, the primary tumors have so much stroma in them that you could not easily see a homozygous deletion. But we saw that 100% of the cell lines had loss of P16 and 90% of the cell lines had loss of P14ARF. So this means that this locus is really critical in mesothelioma. For NF2, we found the first gene ever reported to be repeatedly mutated in mesothelioma, and this was by having good friends. This gentleman here, Andre Klein-Santo, knew this gentleman here from Bristol Myers Squibb. They were both from Argentina. Dr. Klein-Santo put us in touch with Dr. Bianchi, and we started to work together on this. And we ended up finding that 50% of mesotheliomas have NF2 mutations, point mutations. That means just one nucleotide in the gene might be mutated. No deletions. The second copy can be deleted, but what we were finding here was point mutations. And Dr. Knudsen was so excited about this, he wrote a commentary in PNAS saying asbestos and mesothelioma, genetic lessons from a tragedy, our use of asbestos. And of course, me being a person who doesn't mind taking somebody else's title, I learned to take lessons from his title to use for my title today. So what we found here was that 56% of the, we looked at 25 cell lines, confirmed in about 15 of them where we had the tissue available, that the mutations found in the cell lines were also found in the primary tumor. And we found that 14 that had mutations also had loss of expression of NF2 by Western blotting and also had loss of heterozygosity, losing the other copy by loss of all of chromosome 22 where this gene is located. So it made sense, and basically it's fulfilling Knudsen's two-hit hypothesis. So we also studied familial mesothelioma, and you might ask, why? Why look at families? Well, so a little background on that. Generally, only a small percentage of asbestos-exposed individuals develop mesothelioma. However, mesothelioma clustering can be found in some families. By clustering, I mean you might have a family out there that 3 or 4 people in that one family get mesothelioma. You scratch your head and say, how can that happen when there's only 3,000 in the whole country? How could 3 or 4 be in one family? 2 families, W and L, standing for Wisconsin family and a Louisiana family, had multiple cases of mesothelioma and other cancers as well that we initially studied. Neither family was exposed to erionite, which is so carcinogenic, and neither family had any members working occupationally with asbestos. Only trace amounts of asbestos was found in the homes of the W and L family. So our hypothesis was high-risk mesothelioma families like this might harbor a genetic factor that predisposed them either to spontaneous mesotheliomas where no asbestos was involved, or that they were highly susceptible to even low amounts of asbestos. And here's what these families look like. I will point you to, let's see, yeah, all these orange symbols, these are like a family tree. It's called a pedigree. Pedigrees of the Wisconsin family and the Louisiana family. This Wisconsin family has 5 orange symbols. So the circles are females, the squares are males, and this is 1 generation, 2 generations, 3 generations. This mesothelioma is passing from 1 generation to the next, 5 mesotheliomas, and 4 carcinomas, a kidney carcinoma, an ovarian carcinoma, and 2 breast carcinomas. The other family had 7 mesotheliomas, 1, 2, 3, 4, 5, 6, 7. This one is orange and green because this person had in her lifetime 2 cancers, mesothelioma and uveal melanoma, which is a tumor of the melanocytes of the eye. For example, your iris, the colored part of your eye, if you're blue-eyed or whatever, brown-eyed. And 2 people had skin cancers. And I don't want to go into the details, but this is called DNA sequencing, where we look to see if the sequence is identical to what you see in a database. Is it normal or is it abnormal? And in both families, there were different abnormalities of BAP1, the same gene called BAP1. On the Wisconsin family, there was a heterozygous, that means one of the 2 copies had a BAP1 mutation in intron 6. Intron shouldn't mean anything, right? Because that's not the coding region. Exons are the coding. But this is in the splice site of BAP1, and when you have a mutation there, you might not splice out correctly, and you might splice incorrectly and splice out an exon that, when you make your RNA, and therefore you might get, or your DNA rather, and you might end up getting a frame shift and lead to a stop codon that will end up truncating the protein, and that's what happened. This was seen in every person who had mesothelioma or carcinoma in that Wisconsin family. In this L family, this sequence has one abnormality here. So everything to the left and right is fine, except for this one little nucleotide. Here, it causes a nonsense mutation. Instead of a C here, it's a T. Instead of a cytosine, it's a thymine. In exon 16, that was in the germline DNA in the blood. And every affected member of that family had that. So this person, when you have a stop codon there, you don't make the full-length RNA, you don't make the full-length protein, and at the end of the BAP1 protein is a sequence that says, okay, protein, go to the nucleus and do your DNA repair. If you don't have that, it stays in the cytoplasm and gets degraded. BAP1, if it goes to the nucleus, is supposed to cause DNA repair. If it's not there, it's non-functional. And so later we would find that there is a syndrome. It's called the BAP1 tumor predisposition syndrome. And you can look it up in journals now. I even saw it, somebody sent me from China. It was so funny. Somebody sent, one of my former postdocs said, BAP1, blah, 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 blah. I couldn't read anything, of course, it was all in Chinese. Testa, blah, 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 blah. It was really kind of amazing. But what they were describing, one of my fellows on the floor told me that it was saying these very same things, that you can have benign tumors like over here. These tumors are non-malignant. They look almost like pimples, but if you look at them, if you take sections of them and look at under the microscope, the cells are very bizarre looking, but they're not malignant. They can become cutaneous melanomas. Other people can have uveal melanomas. It can be on the iris or it can be in the back of the eye, and they can get really enormous, and they can metastasize to the liver. If they do that, the person dies within a few months. And of course, other people can have like mesotheliomas. This one is a massive one. I've never seen one this big. It's invaded the entire lung. So here, in my final part of my presentation, I want to talk about spontaneous and asbestos-induced tumors in BAP1 mice. We made 3 different kinds of mutant mice, 2 that looked like the Wisconsin family and Louisiana family, and 1 was a conventional knockout mouse. I mentioned these over here. And we looked at the spectrum of spontaneous tumors. That's no asbestos, just do they develop tumors spontaneously just because of the mutation. Well, it turns out about 58% of the mice did develop spontaneous tumors, and most of those tumors were ovarian tumors. 3% were mesotheliomas. You had some breast and lung tumors, but actually it wasn't like the human. We didn't have any uveal melanomas, and so it wasn't exactly the same as in humans, and that's common when you use mouse models. But then we decided we use these mice, we'd use heterozygous mice. These are the ones that have the germline, just 1 point mutation, and we compare them to their normal litter mates that don't have the BAP1 mutation and give them asbestos injected into their peritoneal space. And this is what we found. Mice that had the germline heterozygous mutations of BAP1 have an increased susceptibility to asbestos-induced mesothelioma and a shorter survival. So you can see that the BAP1 mice, the heterozygous plus minus, that's these guys here, they die faster. This is a survival curve. They're dying at about 43 weeks. The wild type mice takes them about 55 weeks, and not all of them get mesothelioma. We also did it with the two clinically relevant, the Wisconsin and Louisiana mice, and the same thing happens. 71% of the Louisiana mice and 74% of the Wisconsin mice develop mesothelioma. Less than half of the wild-type mice develop mesothelioma. And again, the Louisiana and Wisconsin mice survive a lot less. That fell off the chart here is saying it's statistically significant. We also found that if you look at mice that are given asbestos that have either wild type, they don't die very quickly, or if they have P16 heterozygous, or if they have ARF, they get mesothelioma faster. But if you have a cohort with knockout of exon 2 that knocks out both P16 and ARF, they die much quicker and get a higher accelerated rate. So that CDKN2A locus, if you knock out both of those genes, that is the worst thing. They die quicker, they have a higher incidence of mesotheliomas. We also generated, a couple more minutes. We also generated BAP1 conditional knockout mice using a special technology called zinc fingers, which is like nowadays you've heard CRISPR. CRISPR does the same kind of thing. Our facility can knock out this exon, and if we give mice adenovirus that expresses a Cre recombinase, an enzyme that will excise or remove this exon in the mice, we inject it into either the pleural space around the lungs or into the peritoneum. If we do that, we'll lose both copies of BAP1 because we've made the mice homozygous for this mutation. We also got from other investigators NF2, these are called floxed mice because they have a flox or LOXP site on both sides. We can do that for all three of these genes that we think are important in mesothelioma. And then we can knock out 2 copies of these two different genes, any 2 genes we want, or all 3.
Do you put these mice in normal diet, Joe?
Normal diet, yep.
Control.
And then we tested whether these tumor suppressor genes cooperate, and we find that they did. If you have only BAP1 alone, just like in humans, you don't get many mesotheliomas. If you have 2 genes together, you get a little bit more, depending on which genes. If you have all 3 genes together knocked out, you get many more. And if you look at their survival curves, you can see knocking out all 3 genes, this red one, look how fast you get. The median survival is here, the 50th percentile. That is about 12 weeks. So they'll die within 12 weeks without any asbestos, just knocking out those 3 genes. So I'm at the path to cancer again, but this time specifically for mesothelioma. So what is the gene important for mesothelioma at the breakthrough? We think it's BAP1 because it has to do with DNA damage and chromatin remodeling. For expansion, we think it's CDKN2A because it's involved in a cell cycle gene that regulates RB. And we think it's involved in the P53, I lost the P here, P53 checkpoint pathway that's also involved in decisions about whether cells live or die. And then for the invasive phase, John Chernoff at our center, our cancer center director, has been studying NF2 and its role in cell motility, and our lab has studied its role in invasion, so we think it has a big role here. And also AKT, which my lab has studied. AKT regulates many different things, including cell survival and progression of cancers. The last thing I'll mention is about chemoprevention. We're doing studies to use our models. We don't want to just waste our models. We want to use our models to study whether they can be used for chemoprevention. And one of the things we did was we took a drug that's already on the market. You wouldn't have to get a FDA approval. It's called anakinra. It's used for gout. We didn't think it would work in mice because we thought it's a recombinant molecule. We thought it wouldn't cross-react with mouse. We gave it to mice that were treated with asbestos. It's an interleukin or IL-1 beta receptor agonist. It's supposed to reduce inflammation, like if you have gout in your foot or your knee or something. And what we found is that it inhibited asbestos-induced mesothelioma formation in a very, very aggressive model that has NF2 and CDKN2A knocked out. And the delay in tumor onset was due to the inhibition of IL-1 receptor pro-inflammatory signaling. So we had a delay. This is actually the best delay we ever got with a preventive measure. Instead of dying at 22 weeks, they died at 33 weeks. They still got mesothelioma, and the result in proliferation with this marker called Ki67, we greatly reduced the amount of proliferation as well. So we think it has a lot of promise, and if we use this with a less aggressive mouse like BAP1, we think it might really be highly useful. And just recently, this spring, we got a new NCI-funded contract for about $1.5 million to fund a study with, believe it or not, a compound that was derived from broccoli. And this study rationale is because cancers like mesothelioma are so advanced, they become resistant to therapy. So maybe it's a lot better, just don't get the cancer in the first place. Try to prevent it. So for people who are at high risk, like people who have BAP1 germline mutations, or other kinds of germline mutations that predispose them to mesothelioma, or people who work with asbestos, maybe you can give them something. In fact, our surgeon, Joe Friedberg, he takes every day a compound that contains this reagent. And the compound he takes also has an enzyme that activates this sulforaphane even more. And he's been taking it for 6 years. So if you take a preventive agent, it should be something that can't do any harm. Like a doctor says, do no harm. And it's basically like broccoli. And it smells a little bit, but anyway. So what we're doing now, we just got from the company 7,000 bottles, and one of the guys in the lab is using a mortar and pestle, grinding it up, and we're going to then send it to a company that will end up putting it into the feed. So we'll have an adequate concentration. We'll put it in the feed in our laboratory animal facility. Anyway, it's a non-toxic agent with potent antioxidative stress, anti-inflammatory properties, possesses epigenetic modifying properties by its ability to reverse histone and DNA modifications affecting signaling pathways such as those involved in reactive oxygen species and DNA repair. And we'll test it, if it can, eventually if it's successful, we're going to test it in asbestos workers and in BAP1 syndrome patients.
Is that why you're eating so much broccoli at lunch?
Well, you'd have to take, like, you know, a bowl this high to take what you get in one of these pills. But your mother must have had something on her mind when she told you to eat your vegetables and so forth, right? The last slide I have here is to say we also, you know, we do work with humans. We get human specimens from our surgeon, Joe Friedberg. It's a delight to have him. He came, he was originally at Penn. He's one of the best surgeons for mesothelioma in the whole country. We're doing early mesothelioma detection studies on high-risk patients, including asbestos workers, who I'm very fond of. They remind me very much of some of my family members who were working class people who were in construction and did work around a lot of dangerous products, including asbestos. We're also looking at BAP1 mutation carriers, and in about the last 3 years, people have found that there are other, including my lab, that there are other predisposing gene mutations. Most of these other genes that might predispose to mesothelioma are involved with DNA repair, like ATM, BRCA2. Joe knows some of these, and Losby, I'm not sure how many other of you had that kind of background, but any of these genes that are involved with DNA repair, if they're disrupted, then they can't repair if any damage occurs from asbestos, so that might make a person generally susceptible to getting mesothelioma if they're exposed to even very, very low garden variety amounts of asbestos. And what we're doing all the time is serum marker studies, very similar to what people do for serum marker when they do a PSA test for any of you gentlemen. When you get older, you're all so young now, but someday you'll have a prostate and you'll know about it. Right now you don't know about it, but it will enlarge. There's no one in this room who is male who will not have an enlarged prostate. And when your prostate gets larger, you don't want it to become cancerous. And so what they do is they do a PSA test, which is a serum marker test to see if you have, what's your baseline and does it ever elevate? And if it elevates, that could be because of an infection, nothing to worry about. But it might also be because you might have early signs of prostate cancer. So we're trying to do the same thing for mesothelioma patients using this marker, SMRP, soluble mesothelin-related peptides, and this other homeobox group, B1, as markers for that. And we use ELISA assays, which is the simplest thing. I could have any of you come to my lab inside of the first day, you'd be an expert using this piece of equipment. We store samples in these liquid nitrogen tanks down in our biosample repositories. These things are so big that around here, technologists goes up 2 steps to stand and put the samples down into the liquid nitrogen tank. And finally, I'd like to thank the people who are in my current lab, which is kind of small these days. I used to have about a dozen people in the lab. When you get old, it's hard to keep getting grants. We have 2 small grants and 1 large grant now, but there were days when it used to be 3 times this size, and you know, it is what it is. But we work hard as hell, and we're fortunate to get some money from the local 14 International Association of Heat and Frost Insulators and Allied Workers. They stopped, the Allied used to be asbestos workers, but they stopped using that because they thought it would scare people. But we do get donations from them. Every year they have a golf outing to raise money. And then people from other golf outings come and they also bring money. So we've been extraordinarily fortunate, and I think because our heart is really in it, it seems it's contagious and they get into it too. But I want to thank Yuraj Kataria, who does all of our animal studies. He supervises all of that. Nick Rico is new to our lab. I wouldn't put him second, except it's alphabetical. But he is working out really well, and we really like what we saw from him when he was a student in our lab. And we are open to any students from Chestnut Hill who want to come and join us for a summer or during the year. He worked out really well. He's a hard worker, and he's being trained by Eleonora Sementino, who has got to be one of the best technologists I've ever seen in almost 40 years of my career. And Ujwal Shrestha, who is also working with our animals. And Mitchell Chung is trying to get back in my lab. He's working sort of on and off in my lab while he's in another lab. And if I get another grant, we're close. We got 13 percentile, but only funding to 12% gets funded, so we're resubmitting that grant. If we get funded, he may come back to our lab, and maybe we'll get another person as well. So But I think I'm going to try to keep it at no more than about 6 or 7, even if we can get another grant. And these are some of the past postdocs and mostly postdocs and some technologists we've had over the years. Some of these people have gone on to be full professors. Daphne Bell is now chief of the, I forget the name of the section at the National Human Genome Institute. A couple of others are full professors at various places. So I'm very delighted that I've had the good luck of having so many good people. I'll take any questions if you have any. Thank you. The kind doctor.
Joe, thank you so much. What a great talk. I'm fascinated by a lot of aspects of your talk. I'll ask 2 questions. One is, do we have any idea of the prevalence of exposure these days? I mean, in the United States, where it's long been known that asbestos is carcinogenic, so do we have any sense of exposure? And talk a little bit more, if you can, about what, by knowing that these mutations, that in combination, they seem to exponentially increase the likelihood that you'll develop the disease. How is that informing how you're targeting your preventative efforts?
Well, I think in terms of how we're targeting, clearly, I think for prevention, it's probably going to be the same for any of the genes that are involved with DNA repair, and that looks like it's there's about 26 new genes that have been identified. Some of them are, the support is minimal, but I think for any of those, it would be something that involves pathways that might inhibit pathways that have to do with DNA repair. So basically, we want to promote pathways that would counteract defective repair. And so like this SFN, this sulforaphane, that's one of the goals of that. And the first question was about whether the prevalence is really high. I would say that the prevalence of the disease has actually somewhat gone down compared to if you do it based on the number of people in the United States. So we were up around 2,500 cases a year when we were at around 250 million in the United States. And now we're close to 340 million population in the United States, and it's 3,200 a year. So I know this fellow, Michele Carboni, I used to collaborate with, he thinks that the incidence has actually gone down, which may indicate that the protective measures we're taking are less, and that it may have less to do with asbestos and may have more to do with genetic predisposition. Of course, we still need another probably 10 years to determine if that's true, but it may very well be the case that in the United States the incidence will be going down because about a third of all of our affected individuals who develop mesothelioma used to be military. And I would say the exposure of military in the United States is a lot less than before. It's when they go to foreign countries where there's little or no regulations where they're getting exposed. So if they go to Iraq or if they go to other places in the Middle East or India or places like that where they don't have any regulations, and buildings like these buildings you're seeing on TV that are coming down, you can imagine the amount of dust that's being brought up. And even in the United States, when we blew up those buildings, when the buildings were blown up in 9/11, the first building, to the 70th floor, I believe, there was asbestos, like 400 tons of asbestos. Supposedly, the second building did not. Actually, some people think that if we had had asbestos in both buildings all the way to the top, that there would have been less likelihood of the what's the word I want to use? Divergence of the corners of the buildings coming apart if there was asbestos that had, but we stopped at 70 floors, and so you saw that opening up, and then it was like a pancake effect. I don't know if that's really true, but actually the second building came down, it seemed faster than the first, and the second building had supposedly none. But I think in the United States, there are a lot of law cases where I get called to get involved. A lot of lawyers are trying to say it's because somebody has bad genes and it isn't because of asbestos. Right now, it looks like the general number is about 10% of people have a genetic predisposition, but it's not clear if it's a genetic predisposition alone or it's a genetic predisposition to even low levels of asbestos. We just submitted a study, it's under review now, showing that very, very low exposures to chrysotile and even chrysotile, and in fact we were surprised chrysotile was even more than chrysotile, which goes against the grain, and we're going to probably have trouble getting that paper accepted. Even low levels of chrysotile was sufficient in BAP1 mice to cause a high incidence of mesothelioma. So it may be that you'll start to see arguments that, well, how did we know that some people would be so sensitive to mesothelioma? And there are some states like California that have I think it's called the eggshell law, where people, who, for example, who have brittle bone disease, they developed this law because somebody who has brittle bone disease gets hit from the back by another car and suffers severe damage, even if hit by 5 or 10 miles an hour by the person behind them. And people try to say, well, look, I wasn't going that fast. How can that person, sue me for all this money? Well, you're still culpable, at least according to California law, because that person had brittle bone disease, and you're still responsible, is how they've ruled. And the same thing if you have a BAP1 mutation, still the asbestos company, even if it was only low exposure, is out in California, they're responsible. It may not be that way in New Jersey or something, but that's the rule there. And I think that's going to become a national discussion by law firms across the country. So anyway, other questions?
Yes, here I am, Joe. Thank you for coming to Chestnut Hill College. And
That's fine.
you added more prestige to our biomedical lecture series now.
Thank you.
And I congratulate you for taking this colossal project. This is not a small project, mesothelioma, so important. And a lot of people took a lot of message from you. Excellent introduction and all this.
Thank you.
Everybody to take home message from you. You're also working with the familial as well as, are you also working with sporadic cases?
Yes.
Of mesothelioma, both
Yes.
of them. And you have a big uphill battle because of the amount of work you are taking. I wish you all the best with your grant. I know how hard it is to get a project. Now, how do you get all the patient samples for you to work on this project?
Well, for many years, I was writing to people at Mayo Clinic and University of Maryland, where I used to work. I would write to people at Sloan Kettering. I was getting a lot of cell lines from a collaborator at Sloan Kettering. Basically, it was on the phone, and offering, collaborations, and people could be collaborators on papers and things like that. But at some point, when I got to Fox Chase, I said, geez, we're not seeing many patients. What am I going to do? And so that's at one point where I started to do mouse models, because I could at least control and ask the questions I wanted to ask. So a lot of what I did for, I'd say about 15 years was with cell lines and with mouse models. And it's too bad it happened at this stage of my career, but finally we got a really major figure in surgeon in the mesothelioma field, and it was only by accident. We had tried to collaborate. Joe Friedberg used to be the top surgeon for mesotheliomas at Penn, and then what happened was he was more or less discouraged from collaborating because they didn't want samples to be given to another institution. It was politics, and so we never got the samples. And then he eventually took a job down in Maryland. And then we tried to get something going there, and again, there was like, because you're at two institutions, it never happened. And so at one point, he was in Maryland, and his wife was with 3 sets of twins in Philadelphia, and he asked someone at Fox Chase, could he come up here? And he made the play to try to come here, and so we've been having, in fact, right now we're going to be highlighted in this month's cancer letter, which goes around to all the cancer centers in the United States. And so it's so exciting for me to finally have a surgeon I can work with, but let me put it this way, I wish it happened when I was 40.
Congratulations, commendable for all that.
Thank you, thank you.
Joe, I can follow up. Looking at your karyotypes, it's very striking how unstable the genomes are.
Yeah, yes,
Where you're losing entire chromosomes as well as getting translocations.
yes.
One question is, do you have any idea of why that's the case? What about mesothelioma is doing that? And also, do you see that in your mouse models?
Well, we don't see as many rearrangements in the mouse as we do in the human. I'm not sure the reason for that. We also don't see as many rearrangements in the mouse as we see in the human. Now, one wonders, could it be because the mouse has long telomeres and maybe they have greater stability? Mesothelioma is a disease of the aged, and as we age, telomeres erode, and there's a greater chance that you might have two chromosomes being able to fuse and causing a lot of rearrangements between 2 chromosomes. The funny thing about mesothelioma, though, is that despite it having lots of chromosome rearrangements, it is one of the malignancies that has the lowest number of tumor mutation burdens. It has about 1 to 1.5 mutations per megabase, whereas something like melanoma has something like 200, 300 mutations per megabase. So it doesn't have a lot of point mutations, but it has a lot of rearrangements. Yeah, and nobody knows the answer to that. If that's the next question, I don't know why that is. And you would think because it has a low mutation burden, everyone thought, all the physicians thought, whoa, this is going to be a great tumor type for immunotherapy, because immunotherapy works best when you have a low mutation burden. And yet for mesothelioma, it's almost universally a cold tumor, not a hot tumor. So we're trying to find now different ways to make the tumor become hotter by using different drugs to change the phenotype of the tumor microenvironment And for me, this is like getting into a whole new world. And again, what I want to say to anybody who is interested in research is that I could never have done this in the place I was at before, but being at Fox Chase, we have so many investigators that are doing this kind of work that I can see these people now, unfortunately, we have a lot of Zoom meetings, but we can hear all these investigators and go to these other investigators who are doing this work, and suddenly I can become sort of a faux immunologist, either by collaboration or even having people in my own lab start to do this. I mean, wouldn't you say, Eleonora, Nick has become pretty darn good as an immunologist? It's amazing. So if you can get into a really good lab like at one at Fox Chase, you'll learn so many things that'll prepare you for your future if you want to go on in science and medicine. It's just a really good place.
Thank you.
Thank you very much.
I just want to thank you, Dr. Tester, because I had to cancel my cell bio class yesterday to get my PowerPoint prepared. So you actually talked about a lot of things I would have discussed, like exons and splicing and so forth and so on. So that worked out just beautifully.
So this counts.
Yeah, it does. This does count. And the other thing I just want
We're
to
still
say,
cutting your pay.
I also want to say that you may have seen around 3 o'clock students leaving. That's because the student athletes have their field practice. So it wasn't because they weren't interested. It was just because of that dynamic. So thank you again.
Thank you. Well, I must say,